A very warm welcome to all. Today I am going to talk about the diversity of microorganisms. It has been anticipated that our planet Earth is about 4.6 billion years old. Fossil records indicate that prokaryotic cells were present from around 3.5 to 3.8 billion years. Thus, prokaryotic life arose very shortly after the temperature of the Earth decreased. Very likely the earliest prokaryotes were anaerobic. Cyanobacteria and oxygen producing photosynthesis probably developed 2.5 to 3 billion or more years ago. Microbial diversity increased greatly as oxygen became more abundant. It appears likely that modern eukaryotic cells arose from prokaryotes around 1.4 billion years ago. The term biodiversity is used to describe morphological, metabolic or physiological variances among living creatures. The term was coined by W. G. Rosen in 1985. Biodiversity means biological diversity, the field of total variability of life on Earth. Has gained significance after the Earth Summit in 1992 in Rio de Janeiro, Brazil. One of the most fascinating and attractive aspects of the microbial world is its extraordinary diversity. Microbial diversity is comprised of major three interrelated components: genetic, functional or metabolic, and taxonomic diversity. The diversifying factors such as morphology, associative, physiological, and genetic act to establish differentiating characteristics between microbial species. Microbial populations indeed exhibit great diversity in their morphological, physiological, metabolic, and genetic characteristics. Genetic diversity arises due to the dynamic processes of evolution, and functional diversity results as a consequence of genetic diversity and ecological interactions. Estimating the exact number of species of bacteria from any natural ecosystem is impossible with even today's technology. One survey found that there are over 20,000 different species in 1 liter of seawater alone, and nearly 4,000 varieties of organisms in 1 gram of soil sample. Currently, estimates of the total number of species range from about 10 million to a billion, but these estimates are tentative and may be off by many orders of magnitude. Moreover, only a fraction of the species is identified. At present, only 5% of the microbial groups have been described. Our understanding of the role and diversity of microorganisms is limited largely because of our inability to culture more than a few percentage of them. Before moving further, first let me tell you that what is a microbial population, microbial community and niche. A group of microorganisms of the same species that reside in the same place at the same time constitutes a microbial population. A microbial community consists of populations of one species living in association with populations of one or more other species. Niche in ecological theory, the biotic and abiotic characteristics of the microenvironment that contribute to an organism's competitive success. In other words, niche is a habitable ecosystem whose resources and growth conditions are suitable. Individual microbial cells present there will grow to form populations. Microbial diversity can be explained by a variety of approaches like ecological diversity, functional diversity, physiological diversity, genetic diversity, species diversity, and taxonomic diversity. First, we will see ecological diversity. It refers to the number of species in the given area or niche. The species changes within and between the ecosystem owing to changes occurring in composition due to ecological processes and interactions within the environment. This indicates that both biotic and abiotic factors influence the quality and quantity of the species present in the ecosystem. Interactions between these factors results in the composition of the species present in that habitat. Second is functional diversity. It is the expression of microbial diversity based on expression of the metabolic potentials and activities of microorganisms and the microbial community. All species have some differences in their function. Either it is in terms of the presence or absence of a particular function or the intensity of the selected function. As for example, if you take two isolates of E. coli, they may have different rate of glucose utilization depending upon their brought up. Similarly, sometimes these two E. coli differs in term on utilization of substrate. One may utilize a substrate and second may not. This functional diversity will play significant role in various functions carried out by the microorganism, and they may be one of the selection criteria of the culture for the required function under the study condition. Next is physiological diversity. It describes physiological functions of microbial population. This includes the physiological tolerance and preferences of microbes in terms of pH, temperature, salinity, nutritional versatility, etc. Microorganisms develop variations in their pH, temperature, salinity, etc. tolerance and optimum conditions. These variations are mainly due to the adaptability of the organisms and the ecosystem in which they live and do their activity. Due to physiological diversity, we can choose the required strain of the species useful to us. Suppose we want an E. coli which can do best function at acidic pH, we can select the one growing optimally at desired acidic pH. Next is genetic diversity. The genetic variation occurs by various mechanisms within the species that gives rise to diversity. Species are the evolving lineage and the maintenance of their capacity to evolve requires the existence of genetic diversity. Genetic makeup is the main criteria for observed great diversity among microorganisms. In case of bacteria, due to natural selection, there is a possibility of exchange of genetic material through transformation, transduction and conjugation, which result in the development of various new characters in the organisms. Of course, due to various methods of gene transfer in bacteria, sometimes it creates problems in classification and taxonomy of the microorganisms. Next we see species diversity. It is a measure of entropy of the community, that is, an index of diversity measures the degree of uncertainty within a particular microbial community. It is a count of the number of species. In case of microorganisms, methods used to define a species is different from higher living beings. Till date, there are more than one concept for this. The criteria used are not always matching. Next is taxonomic diversity. It is considered to be more indicative of the biodiversity of a habitat than the species diversity. Here the habitat plays a significant role. Now we see ecosystems and microbial diversity. Microorganisms do not live alone in nature, but instead interact with other organisms and with their environment. Therefore, they carry out many essential activities that support all life on earth. Major habitats of microorganisms are soil, freshwater and the oceans. They are present in any habitat containing plants and animals and also within any living creature. Besides these normal habitats like surfaces, biofilm, subsurfaces, rock, sand, etc., they inhabit in hostile environments such as superheated hydrothermal vents, glacial drift materials and deep sea sediments. So, microbes are ubiquitous on Earth's surface and even deep within it. They inhabit boiling hot springs and solid ice, acidic environments near pH 0, saturated brines, environments contaminated with radionuclides and heavy metals, and the interior of the porous rocks that contain little water. Next is diversity in colony characters. Microorganisms are simple in cell makeup. Bacteria are unicellular in spite of these different groups of bacteria. When grow on nutrient medium, they give a variety of colonies. Major variation in colony characters is that they may be transparent, translucent or opaque. The diversity in microbial colony forms varies from punctiform, circular, irregular, filamentous, rhizoidal to even spindle. In terms of texture, they are smooth, rough, mucoid, matte, moist, etc. In terms of color, they produce water soluble or water insoluble pigments with colorless or different shades of red, yellow, orange, black, green, purple, etc. They also show a wide diversity in the colony margin like entire, undulate, lobate, erose, irregular, filamentous, curled and so on. Some microbes form flat dry colonies or with flat raised margin, raised spreading edge, convex, pulvinate, umbonate, moist, gummy or even growth into the medium, partially merged into the medium. Thus, if we want to study biodiversity of microorganisms, it starts from the variety and variability of the colonies they give when they are grown on solid medium. Next is morphological and physiological microbial diversity. One might expect that small, relatively simple organisms like prokaryotes would be uniform in shape and size. Although it is true that many prokaryotes are similar in morphology, there is a remarkable amount of variation due to differences in the genetics and ecology. Microorganisms are quite diverse both in morphology and physiology. They can stain either gram-positive or gram-negative, while like mycoplasmas are devoid of cell wall. Microbes are found in diverse shapes and size like spherical, straight or curved rod-shaped, vibrios, spiral, lobed, oval, pear-shaped, plate-shaped, flat square-shaped, helices or filaments, irregularly shaped or pleomorphic that are variable in shape. They can exist as individual cells, in pairs, in long chains, clusters, tetrads like cubical packets, long multinucleate filaments or hyphae that may branch to produce a network called a mycelium or aggregates. They may be having sheaths or capsules, endospore or spores on hyphae. Microbes vary in size as much as in shape, as they range in diameter from 0.1 to over 15 micrometer, and some filaments can grow up to 200 micrometer in length. A rod of about average size is 1.1 to 1.5 micrometer wide by 2 to 6 micrometer long. A few bacteria reach 500 micrometer in length. Bacteria show true motility with flagella or can be non-motile. Flagellation can be diverse like peritrichous, amphitrichous, lophotrichous, monotrichous, or while some show axial filament and even gliding motility. Diversity in appendages is found due to the presence of pili and fimbria, prostheca or stacks. Reproduction may be by binary fission, budding, fragmentation or other mechanisms. Diverse physiology recorded as microbes can be aerobic, facultatively anaerobic, microaerophilic or strictly anaerobic. Some are mesophiles, grow in the range of 15 degrees centigrade to 45 degrees centigrade. Psychrophiles that grow in the range of -20 degrees centigrade to +10 degrees centigrade, while others are hyperthermophiles, which can grow above 100 degrees centigrade, that are found in extreme aquatic and terrestrial habitats. Microorganisms are present in fresh water, ocean water having 3.5% salt to hypersaline environments having 34% salinity. Many are symbionts of plants or in animal digestive systems. Now, next is metabolic diversity. Nutritionally, microorganisms are highly diverse as compared to any other life forms on earth. They show great versatility in metabolism. Principally for carbon source, all microorganisms show autotrophic, heterotrophic or mixotrophic metabolism for synthesizing cell mass. That is, carbon is obtained from carbon dioxide or from organic compounds, or from both organic compounds and by fixing carbon dioxide. Secondly, microbes show diversity in a wide range of electron donors, that is, obtains reducing equivalents from inorganic compounds, that is lithotrophic, or organic compounds, that is organotrophic. Moreover, microbes can obtain energy from external chemical compounds, that is chemotrophic, or from light, that is phototrophic. Not surprisingly, in view of the variety of their lifestyles, microbial metabolism varies greatly between the members of different groups. Some are organotrophs, others are autotrophic or phototrophs. In practice, these terms are almost freely combined. For instance, chemolitho-autotrophs obtain energy from the oxidation of inorganic compounds and carbon from the fixation of carbon dioxide. Examples are nitrifying bacteria, sulfur oxidizing bacteria, iron oxidizing bacteria. Likewise, other groups are photolitho-autotrophs, chemolitho-heterotrophs, chemo-organo-heterotrophs or photo-organo-heterotrophs. In short, the microbial world exhibits a great variety of processes, aerobic as well as anaerobic, phototrophic, that is oxygenic and anoxygenic, respiratory, that is with oxygen or other electron acceptors, fermentative and chemoautotrophic, that is using reduced inorganic compounds as energy sources. Finally, sequencing of microbial DNA isolated from natural environment has revealed the existence of a tremendous variety of yet uncultured microorganisms, showing that the true microbial diversity in nature is much higher than currently recognized on the basis of organisms studied in culture. The variety and variability among microorganisms we know today is based on less than 10% of organisms, as nearly 90% organisms are till date not cultivated. Thus, we have no knowledge of these 90% of the microbial population. It is very essential to know microbial diversity and understand to conserve it, because we all want to live a healthy life. This is the only possible if the diversity is not lost and wisely protected, because all higher living beings have several microorganisms in their body as normal flora of the body, and in the absence of them we cannot do all metabolic functions necessary for our own healthy life. Thank you.
Let us summarize the topic. Prokaryotic and eukaryotic microorganisms dominate life on earth with respect to the number of individuals and biomass. Yet, the number of species of macroorganisms described as well as the estimated numbers of yet to be described species surpasses those of the microorganisms by an order of magnitude. Such numbers give an incomplete picture of the diversity in the microbial world. Only two modes of life are found in higher plants and animals: oxygenic photosynthesis and aerobic respiration. In the microbial world, a great variety of processes occur, aerobic as well as anaerobic, phototrophic in presence or absence of oxygen, respiration with oxygen or other electron acceptors, fermentative and chemoautotrophic metabolism. Also, phylogenetically the microbial world is by far most diverse. The microbial world encompasses most of their phylogenetic diversity on Earth, as all bacteria, all the archaea and most lineages of the eukarya are microorganisms. The morphological diversity in the microbial world is much smaller than that of the higher plants and animals. With respect to number of individual microorganisms and the biomass, the microbial world is far more important than the world of macroorganisms. But the number of species of microorganisms described to date and the predicted number of yet undescribed species are about an order of magnitude lower than those of the macroorganisms. Based on their modes of energy generation and variety of organic and inorganic compounds used as energy source or as nutrients, the microbial world and especially the world of the prokaryotes has many more possibilities than are realized by the macroscopic animals and plants. Thank you.